Good morning, children. Today we are going to discuss about male and female reproductive parts of plants. The male reproductive parts, that's called as a anther. Structure of anther. Anther are classified into epidermis, archisporium. Archisporium divided into parietal tissues and sporogenous tissues. Parietal tissues are divided into three layers: endothecium, middle layer, and tapetum. Sporogenous cells are divided into pollen mother cell, pollen tetrad, and pollen. In a flower, stamen is considered as the male reproductive organ. Each stamen consists of filament, connectivity, and anther. Anther may be monothecous or dithecous. A monothecous anther consists of two locules or two sporangia, so it is said to be bilocular or bisporangiate. A dithecous anther consists of four locules or four sporangia, so it is said to be tetralocular or tetrasporangiate. Development of microsporangium is eusporangiate. Sporangium developing from a group of cells. A very young anther in transverse section show epidermis and archisporium. The archisporial cells divide periclinally, gives rise to primary parietal cells on the outer side and sporogenous cells toward inner side. The cells of the parietal layer divided periclinally and anticlinally forms endothecium, middle layers, and tapetum. The cells of the primary sporogenous tissues are differentiated into pollen mother cells or microspore mother cells. The anther wall consists of following wall layers: epidermis, endothecium, middle layers, and tapetum. Epidermis. Epidermis is the outermost single layer. It is compactly arranged and usually protective in function. Epidermal stomata was reported in Alangium. Endothecium. The cells of endothecium are readily elongated and show fibrous bands. The fibrous bands are made up of callus and arise from the inner tangential walls. Usually, fibrous bands are U-shaped. The fibrous bands are hygroscopic in nature. Endothelial cells helps in the dehiscence of anther at maturity. Because of the presence of fibrous band, this layer is otherwise called fibrous layer. It is usually single layered, exception coccinea double layer. Middle layer. Below the endothecium, 2 to 3 layers of cells are present which constitute middle layer. These layers are ephemeral and become crushed by early meiosis in pollen mother cells. These cells act as storage centers for starch. Tapetum. Tapetum is the innermost layer of anther walls and is completely surround the sporogenous tissues. The cells contain dense cytoplasm with prominent nuclei. Usually, tapetum consists of single layer of cell. As the tapetum completely surrounded the sporogenous tissue, major part of it is derived from the parietal cell and a small part developed from the sporogenous tissue. Tapetum transport the nutrients to the developing sporocyte. Tapetal cells are opening and it is red-brown in apple or violet in anemone. On the basis of behavior, there are two kinds of tapetum. First one is amoeboid tapetum. The inner and radial walls of the tapetum break down due to the action of hydrolytic enzymes and their protoplast penetrates between the pollen mother cell and developing pollen grains. After insertion, they fuse with each other and forms a mass of tapetal periplasmodium. This tapetal plasmodium remain associated with the pollen grains till their maturity. When the anthers get drying up the tapetal, periplasmodium gets dehydrated and coated over the surface of pollen grains, thereby helping in the formation of exit. Amoeboid tapetum is considered as a primitive type. It is also called periplasmodial tapetum. Example: Alisma, Tradescantia, Typha, Sagittaria, just like that. And another one, glandular tapetum. Glandular tapetum. The cells of glandular tapetum remain intact throughout microspore development. They secrete their substance from their inner face. Secretory tapetal cells are thin and possess almost all cell organelles like mitochondria, plastids, dictyosome, etc. Just before the pollen mother cells undergo meiosis, the walls of the tapetal cells become thick and there is considerable increase in the number of ribosomes and pro-Bush bodies. With the completion of pollen development, pro-Bush bodies pass into the anther locule from the tapetal cell and they are now called Bush body and they coated over the pollen grains. Example: higher monocots and many dicots. Function of tapetum. The nutrients are transported through tapetum to the sporogenous tissue. Tapetum is involved in the synthesis of callose, which release microspore in a tetrad by degrading cellulose wall. Tapetum play an important role in the formation of enzyme. Pollen kit, lipids and carotenoids is formed by a tapetal layer. It is an insect attractant and protect pollen from ultraviolet. See the figure. The transverse section of tetrasporangiate anther showing various tissues. See the picture for the central vascular strand is there. The outermost layer called as a epidermis. The stomium is breaking region. The presence for tetrad, the microspore mother cells is presented, and middle layer also presented. The breaking of the stomium and bursted the anther wall and pollen grains is released. Microspores. Microspore, that is the pollen grains, are developed inside the microsporangia. The microsporangia are developed inside the corner of the four-lobed anther. Young anther are more or less oblong in shape in a section and made up of homogeneous mass of meristematic cells without intracellular space. With further development, the anther becomes four-lobed. The outer layer of anther is called epidermis. Below the epidermis, at each corner, some cells become differentiated from others by their dense protoplasm. Archisporium or archisporial cells. Each archisporial cell then divides mitotically and forms an outer primary parietal cell and an inner primary sporogenous cell. The outer primary parietal cells form primary parietal cell layer at each corner. Below the parietal cell layer, the primary sporogenous cells remain in groups, that is, the sporogenous tissues, the cells of primary parietal layer, then divided in both periclinally and anticlinally and form multi-layer antheridial wall. The innermost layer of antheridial wall, which remain in close contact with the sporogenous tissue, function as nutritive layers called tapetum. The primary sporogenous cells either directly function as spore mother cells or divide mitotically into a number of cells which function as spore mother cells. The spore mother cell undergo meiotically division and gives rise to 4 microspores arranged tetrahedrally. See this picture? Different stages of development of microspore from microspore mother cell. First one is a microspore mother cell. First division, that's called as a dyad stage. The third picture, the tetrad stage. The fourth picture, cleavage of protoplasm and formation of pollen grains. The last picture, 4 microspore, that's pollen grains with exine and intine. Structure of microspore, example pollen grains. Microspore, that the pollen grain, is the first cell of the male gametophyte which contains only one haploid nucleus. These are the various shapes: polyhedral, milky thistle of Asteraceae, cuboidal for Bacilla alba, trigonal common in Onageraceae, cylindrical in Commandaceae, etc. The size of the pollen grains generally vary from 10 to 80 micron, but the size may be even 100 micron in diameter. The pollen grains have two wall, outer exine. The exine is further differentiated into two regions, outer sexine and inner nexine and inner intine. The exine is cut inside and through with different ornamentations. It may be warty, spiny. It can protect the pollen from the external injury. The intine is very thin, elastic, and delicate. The structure of different pollen grains is given in figure. See the picture? Usually, the mature pollens are not attached in tetrad and they get separated from one another. In some plants, like Typha angusta of Typhaceae, they do not get separated from one another. Compound pollen grains. In orchids and members of Calotropis, Asclepiadaceae, all the pollen grains within each pollen sac remain united, forming the structure called pollinium. The pollen grains of Pinaceae are provided with a two-winged like expansion of exine which help in wind dispersal of pollen. See the pollinium of different plants. Microgametogenesis, development of male gametophyte. Microspore, that is the pollen grain, is the first cell of the male gametophyte which contain only one haploid nucleus. During early stage of development, it remains within the microsporangium. The cell undergoes unequal division and forms a several generative cell and a large vegetative or tube cell. Initially, the generative cells remain laying at one corner of the spore wall. Within short time, it gets detached and become ellipsoid or fusiform in shape and remain suspended in cytoplasm of the vegetative cell, two cell stage, that is vegetative cell and generative cells. Later on, the generative cells is divided and gives rise to two ellipsoidal or lenticular or spherical cell, the male gametes, three cell stage, vegetative cell and two male gametes. See the picture? The pollen grains is there, germ pores is there, and nucleus is the center. The large vegetative cells is there, small generative cells is there. It form the tube nucleus. It formation of a pollen tube and two male gamete and one pollen tube nucleus. So the second division, the division of generative cell may take place either in the pollen grain or in the pollen tube which develop through germ pore affect pollination. The nucleus of the vegetative cell is commonly known as the tube nucleus. It usually shows signs of degenerate within the maturation of generative cell. Finally, the tube nucleus remain within spore or many enter the pollen tube. Sooner or later, it may be degenerated completely. The significance of tube nucleus. Earlier work through that the tube nucleus had great significance in the direction of growth of pollen tube as it is usually present just behind the growing point within the tube, within the pollen tube. Female reproductive system. Stigma, style, ovary and ovule. The flower. The flower is the reproductive unit of some plants. In angiosperm, part of the flower include petal, sepals, one or more carpels, the female reproductive organs, and stamen, the male reproductive organs. The female reproductive organs. The pistil is the collective them for the carpels. Each carpel include an ovary where the ovule are produced. Ovules are the female reproductive cells, the eggs, a style, a tube on top of the ovary, and a stigma which receive the pollen during fertilization. Types of flowers. Some flowers called perfect flower have both male and female reproductive organs. Some flowers called imperfect flowers have only male reproductive organs or only female reproductive organs. Some plants have both male and female flowers, while others have males on one plant and females on other plants. Completely, flowers have stamens, a pistil, petals, and sepal. Incomplete flower lack one of these parts. Carpel. Carpel is the female reproductive organ and is present in the center of plant. The shape of carpel is like a flask. Carpel is made of three parts: stigma, style, and ovary. The top part of carpel is called stigma. The stigma is sticky and receive pollen from anther of stamen. The pollen grains stick to the stigma. The center part of carpel is called style. Style is a tube which connects stigma to ovary. The bottom part of carpel, which is swollen, is called ovary. It is here that the ovules are made and stored. There are many ovules in the ovary, and each ovule consists one female gamete of the plant. The female gamete of the plant, which is present inside the ovule, is called egg or ovum. Therefore, female gametes are made in the ovary of carpel. The female organ of a plant is also called pistil. Also, the carpel is surrounded by a number of stamens. See the picture. The stigma, style, and ovary is called carpel. The female gametes, that's called as a egg at the center. It's covering for ovule. Carpel, female reproductive organs of a plant. Carpel is also known as pistil. There are flowers which are called unisexual. This is because they have either stamen or carpel. Flowers of papaya and watermelon are example of unisexual flower. And the flowers which have both male and female sex organ are called bisexual. Flowers of hibiscus and mustard plant are called bisexual flower. Types of ovules. There are 6 types of ovules which are formed in angiosperm, and the most common type in anotropous ovule. Anotropous ovule. In this type, the body of the ovule became completely inverted during the development. Such the micropylar lie very close to the hilum. For example, gamopetalae members. Orthotropous ovule. The body of the ovule is erect and straight such that hilum, chalaza, and micropyle lie in the straight line. For example, polygonum. Hemitropous ovule. The body of ovule is placed transversely at right angle to the funicle. The micropyle and cell lie in one straight line. For example, ranunculus. Campylotropous ovule. The body of ovule is covered and bent round so that the micropyle and cell do not lie the same straight line. For example, leguminaceae. Amphitropous ovule. The curvature of the ovule is very much pronounced and the embryo sac also become curved. For example, Butamaceae. Cernotropous ovule. The nucleus and the axis are in the same straight line in the beginning, but due to a rapid growth on one side, the ovule becomes anotropous. For example, Opuntia. See the part of pistil, thalamus, ovary, ovules, style and stigma. See the picture. A. Orthotropous, B. Anotropous, C. Hemitropous, D. Campylotropous, E. Amphitropous, F. Cernotropous. See the picture. The microspore mother cells is divided meiosis. The meiosis 2, the megaspore getting for tetrad. Megaspore 3 are degenerated. Surviving megaspore only one. Degenerated megaspore, upper and aposporous initial cell. See the picture. The microspore mother cell, ovule is produced. The embryo sac maintain physical contact with the parent. Sporophyte throughout their development. This association of the female gametophyte and the sporophyte provide an opportunity to examine interaction between the cells, tissues and genomes. See the various stages is occur. Matured embryo sac. A typical matured embryo sac of angiosperm is a 7-celled and 8-nucleate structure. At the micropylar end, an egg apparatus is present which consists of an egg cell and 2 synergid cells. Synergids contain filiform apparatus which guides the pollen tube entry into the embryo sac during fertilization. At the chalazal end, 3 antipodal cells are present. In the center, 2 polar nuclei are present which get fused prior to fertilization to form a diploid secondary nucleus. Hence, 8 nuclei are constituted with 7 cells. Thus, embryo sac is 7-celled and 8-nucleate. See the picture. The chalazal end, the antipodal getting 3, the polar nuclei for 2, the central cell there, the egg 1 and 2 synergids and 2 filiform apparatus, the micropylar end. Filiform apparatus. In the ovule, the female gametophyte, that is the embryo sac is present. Inside the embryo sac, 3 antipodal cells, 2 synergids and central polar nuclei and an egg is present. Inside the synergids, filiform apparatus is present. A mode of pollen tube entry into ovule. Pollen tube eventually reach the micropyle of an ovule, and for this purpose may follow one of the following 3 routes. Porogamy. When the pollen tube enter the ovule through micropyle, it is common for most of the organisms. Mesogamy. When the pollen tube enter through the integuments, example cucurbita, or through funicle. Chalazogamy. When the pollen tube enter through the chalazal tissue, example casuarina. See the picture. The porogamy, chalazogamy and mesogamy. In order to make a new seed male gamete present the pollen grain unit, with the female gamete present in the ovule. This process takes place in two steps: pollination and fertilization. Pollination, when the pollen grains from the anther are transferred to the stigma of a carpel, is called pollination. This is important because it is due to pollination that male gametes are able to combine with the female gamete. Pollination is done by insects like bees, butterflies, and birds, wind and water. There are two types of pollination: self-pollination and cross-pollination. When pollen grains from one flower carried to the stigma of the same flower or to another flower of the same plant, it is called self-pollination. And when pollen grains from a flower of one plant are carried to the stigma of a flower of another similar plant, it is called cross-pollination. See the picture: self-pollination, the same plant; cross-pollination is interchanging for the pollen grains. Insect help in pollination. This happens when an insect site of a flower of one plant to suck nectar, then the pollen grains from another stick to its body. Now when this insect flies and sits on the flower of another similar plant, then the pollen grains are transferred and they sticky to the stigma of the flower of another plant. This way insect help cause cross-pollination. Wind too helps the cross-pollination. Depending upon the source of pollen grain, pollination is the three types: autogamy, that's called as self-pollination. It is the kind of pollination in which of the pollen from the anther of a flower is transferred to the stigma of the same flower: wheat, rice, and peas. Autogamy is further classified as cleistogamy, homogamy. Jaentogamy, the another one type. Xenogamy, for example, for cross-pollination. Agents of pollination. The agent responsible for pollination in angiosperm have been grouped into two main categories: abiotic and biotic agent. Abiotic means wind pollination and water pollination. Biotic agents mean animals, mammals, reptiles, birds, and insects. Adaptation of wind pollination. Wind pollination is also termed anemophily and takes place through the wind. Flowers are small, colorless, inconspicuous, nectarless, and become arranged as inflorescence. The anther are well exposed for the easy dispersible of pollen grains. Pollen grains are small, light, dry, dusty, non-sticky, and sometimes even winged. The stigma are large, hairy, and feathery or branched to catch the airborne pollen grains. Common example of wind pollination flowers are grasses, sugarcane, bamboo, and coconut. Adaptive of water pollination. Water pollination is also termed as hydrophily, and mode of pollination is water. It is quite rare in flowering plants and is limited to about 30 genera, mostly monocotyledons. It is very common in plant groups such as algae, bryophytes, and pteridophyte. Flowers are small, colorless, inconspicuous, odorless, and nectarless, and pollen grains or stigma are generally unventable. The stigma are long and sticky: Vallisneria, Hydrilla, and Zoosteria. Not all aquatic plants has the water for pollination. In a majority of aquatic plants, the flowers emerge above the level of water and are pollinated by insect or winds, and in land plants. example water hazel and lily. In Vallisneria, the female flower reaches the surface of water by a long stalk and pollen grains are released on the surface of water. They are then carried by the passive water current. In most of the water pollination species, pollen grains are protected by mucilaginous covering. Adaptation of insect pollination. Insect pollination is also termed as entomophily. Insect pollinated flowers are large, colorful, fragrant, and rich in nectars. A number of flowers are clustered into the inflorescence to make them conspicuous. Flowers have a nectar gland and they are highly fragrant to attract insects. The surface of the pollen grains is sticky due to the exine layer and the stigma is sticky due to the mucilaginous layer. Nectar and pollen grains are floral, a reward for the insect pollinations. In some species, floral reward are to provide safe place to lay eggs. Example for the tallest flower of Amorphous. In plant, yucca, moth and the plant cannot complete their life cycle without each other. The moth deposits its egg in the lacule of the ovary and the flower. In turn, plant gets pollinated by the moth. The larvae of the moth come out of egg and the seeds started developing. Outbreeding device. Flowering plants have developed many devices to discharge self-pollination and encourage cross-pollination because the majority of flowering plants produce hermaphroditic flowers and are likely to come in contact with the stigma of the same flower. The continuous self-pollination lead to changes of inbreeding device to prevent inbreedings are repetitive of pollen release and stigma is not synchronized. Either the pollen is released before the stigma become receptive or stigma become receptive before the release of pollen. In some of other species, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower, which will prevent autogamy. Self-incompatibility is the third device to prevent breeding, which is genetic phenomena of inhibiting pollen tube growth of the stigma of the flower. Another device to avoid self-pollination is to produce unisexual flower, that is autogamy is prevent if both male and female flowers are present on same plant. Castor and maize, both autogamy and zytogamy is present in several species. Like papaya, if male and female flowers are present on different plants, each plant is either male or female dioecious. Fertilization. After pollination, next step is fertilization. In this step, male gametes present in pollen grains join with the female gametes present in ovule. When the pollen grains fall on stigma in burst open the pollen tube, grow which move through the style towards the ovary and enter ovule. Male gametes move down through the pollen tube. The tip of pollen tube bursts open in the ovule and the male gamete come out. In the ovule, male gamete combines with the nucleus of the female gamete and fertilized egg is formed. This fertilized egg is called zygote. Post-fertilization event. In the plant, fertilization is defined as the fusion of male and female gamete to develop into the diploid zygote. After fertilization, a series of events occur in the zygote to develop into the seed. Let us take an overview of this process of post-fertilization, endosperm and embryo formation. What is post-fertilization? Post-fertilization is a series of events that take place after fertilization to develop a seed from an ovule and the fruit from the ovary. The following event occur in the post-fertilization: endosperm development and embryogeny. Endosperm is the type of tissue which is present in the seed of flowering plants. During the time of fertilization, reserved food material fill in the cell of endosperm tissue. It provides nutrients to the developing embryo into the formation of starch. Endosperm development is classified into three types. These are follows: nuclear endosperm formation, cellular endosperm formation, helobial endosperm formation. During seed maturation or in the mature seed, the developing embryo may either utilize the endosperm completely or it is used by seed during seed germination. Embryogeny. 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 Embryogeny is defined as the process of growth and development of an embryo from a zygote in the flowering plants. Embryo development stage are the same in both monocot and dicot plants. In dicot plants, embryo and embryonal axis and two cotyledons are present. Two parts are present in the embryonal axis. These are follows: epicotyle and hypocotyle. A monocot plant, embryo consists of only one cotyledons. The cotyledon is termed as succulum. In the grass family, root cap of grass is covered with an undifferentiated sheath which is called coleorhiza. A portion of embryonic axis which is called located above the succulum is called as epicotyle. Epicotyle consists of shoot apex and coleoptiles. Formation of fruits and seeds. In the ovule, the fertilized eggs divided several times to form the embryo. A tough coat is developed around the ovule and is gradually develops into a seed. The ovary or flower develops to become a fruit which contains seeds inside it. Other part of the flower like sepal, stamen, stigma, style become dry and fall off. Fruit take place of the flower. Seed is protected by the fruit. Some fruits are soft and juicy while the other fruits are hard and dry. A fruit contains seed of plants inside. The seed is reproductive unit of plant. With the seed, new plant can be grown as a seed contains baby plant and food for the baby plant inside it. The part of the baby plant is seed which grow into the leaves is called plumule, and the part which develops into the root is called radicle. The part of the seed which stored food for the baby plant is called cotyledon. The baby plant inside the seed is in the dormant state. It is only when the provide it with a suitable environment like water, air, light, it germinate and a new plant grows. Wheat grains, gram, corn, pea, beans, etc. Example for seed. See the picture. A bean seed cut open. Seed germination. The seed obtained from the plant or the dry in dormant state. It is only they get water, air, soil. Then they begin to grow into a new plant. The beginning of growth of the seed is called germination of seed. The germination of seed being when they absorb water, swell and burst through the seed coat. It is with the help of water the enzyme function in the seed. The enzyme digests stored food and make it soluble. While the help of soluble food, a radicle and plumule grow. See the picture. Seed germination under suitable condition to produce new plant. These pictures show the germination of a bean seed to form a new bean plant. First, the radicle of the seed grow to form root. This root grow inside soil and absorb water and mineral from the soil. After this plumule grow upwards and shoot are formed. This shoot develops leaves. the leaves being to make food with the process of photosynthesis, and gradually a whole new plant is developed. Thank you.